lejos se me van todos, por Dios. Yo hice la tesis con Juan, sí.
Pero aquí,
En el Roca. En el Roca, bueno, hice también el IBS, en Grenoble, pero vamos.
Buenos días. Good morning. Voy a presentar a Rafa muy brevemente. Hoy nos visita Rafa. Voy a hacer la presentación en español, pero bueno, el seminario va a ser en inglés, ¿vale? Bueno, muy brevemente, la gran mayoría de vosotros ya conocéis su trayectoria, pero es muy conocido en la casa. Rafa creció y se desarrolló aquí en el instituto. En el 2009 obtuvo su doctorado en bioquímica y biología molecular bajo la supervisión de Juan Hermoso. Después hizo una estancia postdoctoral larga en el CENIO con Guillermo Montoya hasta el 2015. De ahí entiendo que tuvo un impasse hasta que finalmente ya en 2018, bueno, impasse quiero decir que volvió a trabajar con Juan. En el 2018 ya se fue a Copenhague, una estancia de 4 años en la Fundación Novo Nordisk. Y desde principios de este año es flamante Ramón y Cajal del instituto. Entre otros méritos, bueno, tiene más de 20 años de experiencia en biología estructural, con más de 40 papers, más de 70 estructuras depositadas en el PDB. Varios proyectos como IP, proyectos competitivos, como un proyecto concedido en la última convocatoria del proyectos de generación de conocimiento y colaboraciones con empresas, lo cual es destacable también. Y bueno, con respecto a sus líneas de investigación, Rafa está muy interesado y especializado en cristalografía de rayos X, microscopía crioelectrónica, para diseccionar maquinarias macromoleculares. En concreto, estudia el interplay entre las defensas antivíricas procarióticas y, desde un punto de vista estructural, para tratar de establecer bases para el desarrollo de terapias basadas en fagos y nuevas herramientas biotecnológicas. Así que, Rafa, bienvenido, gracias.
Bueno, no sé si me oís, creo que sí, ¿verdad? Bueno, muchas gracias, Javier, por la introducción. Bueno, si me alejo creo que no me vais a oír. Bien, bueno, he titulado la charla, voy a explicar, sorry, I'm going to change it to English. So the title, the reasoning behind it, the title of the talk is because, well, I want to give you 2 different ideas. So the first of them is, I want to talk about CRISPR. What is CRISPR? Why is important CRISPR? And where we are right now in the CRISPR technology. And secondly, I want to be focused, so I will move on into my work related with the CRISPR-Cas system 3, which is a crucial role. And this CRISPR-Cas type 3 is, most of the processes I will show you later on the slides are regulated by the presence of a signaling molecule, which is a ring molecule. So that's the idea about this title of the talk today. So what is CRISPR? So I guess most of you in the audience know or have heard about this CRISPR revolutionary technique for genome editing. But basically, these CRISPR-Cas systems, from biology point of view, they are adaptive immunity system from bacteria and from archaea, and is the way they defend themselves against the invasion of viruses and plasmids. And is the only known adaptive immunity system in bacteria, if we compare it with the case of the eukaryotic one, where there are many other adaptive immunity systems. And the classical pathway for this adaptive immunity system, this is the 3 steps of the adaptive immunity system in CRISPR-Cas, is that it follows these 3 steps. So the first one is the adaptation step. This adaptation step consists in the fact that there is a first infection from the virus, and this external nucleic acid is inserted into the bacterial cell, and this insertion, it is recorded in what is called a CRISPR array. This CRISPR array is a way to save a memory of the previous infection in the bacterial cell. So after this initial step of the adaptation, incorporating the external nucleic acid, there is another step which is called expression, and the expression step is where this CRISPR array is transcribed and produce the CRISPR RNA, and then it's also transcribed the Cas protein associated to this CRISPR RNA. So then there is a processing and assembly of these Cas proteins along with the CRISPR RNA for producing what is called the effector complex or the interferon complex. So in the third step, in this interferon phase, is when there is a new infection from the phages, from unknown phages, and the bacteria recognize the previous infection by being detected by the presence of this effector complex and by a hybridization of the external nucleic acid. And then is when the bacteria is triggering the bacterial immunity. So this is what is CRISPR. And there is a classification on the CRISPR-Cas system. They are basically classified depending on the nature of the interference complex or the effector complex. And when the interferon complex, so this mixture between the protein and the RNA that triggers the bacterial immunity, is composed by a multi-subunit effector complex, like in this case, this is when we are talking about the class 1. But when the effector complex is composed by a single protein effector module plus the RNA, is then when we are talking about the class 2. So probably you have heard about this protein like Cas9 or CPF1, which are the classical one used for biotechnological applications. And this is why it is those two ones, especially the class 2, are the ones that are best well characterized biochemically and mechanistically speaking. So this is what is CRISPR, and why is important CRISPR? So CRISPR is important because it can be used as a molecular scissor for genome editing, and which is the classical strategy we are following for gene therapy. So the classical strategy is to use this molecular scissor. So it's basically, there is a protein which is recognizing specifically certain region of the DNA. Then it acts, this molecular scissor is cleaving the protein, the DNA, and then it's producing this double-strand break. And this double-strand break is when it is repaired by the host machinery, by different pathways, and this is when the DNA is repaired. So this is the basis of gene therapy using this molecular scissor. And this molecular scissor, now the most advanced ones is this CRISPR-Cas systems, but it has been developed since the 19 different types of molecular scissors. The first one, it was the thin finger nucleases. But the problem in this case, it was not really easy to redefine, and also we have to fuse nucleases domain for acting as a molecular scissor. Then in the 2000, it came out the other scaffold called meganucleases. These meganucleases are containing in the same scaffold the possibility to cleave the DNA, and also to we can redesign this protein for recognizing specifically a different sequence of DNA. But it was really expensive to modify, to alter these meganucleases for recognizing the sequence we want to use for our gene therapy applications. Then in 2010, it came out the TALEN proteins. This TALEN protein came from bacteria also, and they contain DNA binding domains that we have to fuse with nucleases. They were more specific and more easy to modify, but then also we have to fuse with nuclease domains for acting as a molecular scissor. And then in 2012, it came out this, the most advanced genome editing tool, which is this CRISPR-Cas systems. So the difference between these three first one and the last one is basically that if we want to recognize a specific DNA sequence, we have to modify the scaffold. But in the case of the CRISPR-Cas system, we don't have to modify the scaffold, because the Cas9, CPF1, all these proteins are exactly the same, and what we have to do for recognize a different DNA is to exchange a piece of RNA, which is the one that's going to recognize by hybridization the DNA. So that's why I'm saying here that this CRISPR-Cas system has democratized the genome engineering, because it can be redesigned easily, and they are inexpensive to access in comparison with the other three ones. And actually, in 2020, it was given it was awarded the Nobel Prize in Chemistry to the labs of Emmanuel Charpentier and Jennifer Doudna, in fact, due to the development of this method for genome editing. It was based in one of these protein called Cas9. And here I want to highlight the fact that this RNA is the responsible of the interaction with a specific DNA sequence we want to cleave and to repair. And so since 2012, it was the first application. It was to use directly for editing the DNA, but it has been developed along the time different variation of this CRISPR-Cas9 for doing different functions. Like, for example, we can fuse the Cas9 recognition with methylase domains, and then we can act at level of epigenetic regulation, or even we can fuse peroxisomes for, peroxidases for modifying the nucleosome, so involving in the protein profiling. We can also use it for transcriptional regulation, and these are the last resort of molecular scissors, because in this case, the strategy followed is similar to this one, but following a different pathway from the host cell machinery, and that's why it's more precise, using this base editing and prime editing modifications. So that's why I claim that I think we are surfing in the CRISPR-Cas era, because if we see here, for example, these trends in genomic editing used in clinical trials, we can see that basically since 2016, most of the genome editor tools are based on CRISPR, this Cas9. Sorry, here the Cas9 is a CRISPR-Cas, and it has been even removed, the initial one, like thin fingers. But okay, the application of the CRISPR-Cas is not only the obvious one in biomedicine for gene surgery or for drug development. It can be also used for biotechnology, because it can produce new fuel from modifying bacteria for producing plastic, for example, or even we can modify plants for making them more resistant for drugs, and even we can use this system for producing new materials. But also it's very important for biology, because we can use this CRISPR-Cas system for modifying animal models, and then to study, for example, a specific disease in these animal models. Even modifying genetic or making genetic variation in different species for our purposes. So to conclude, this system, CRISPR-Cas system, has constituted a revolution in all of these fields. And just to illustrate what I was mentioning before, this is new that it was sent like 2 weeks ago. It was in 16th of November. And this is the first case it was used, the CRISPR-Cas treatment, or is the first time it was approved from the UK agency drug, a treatment using CRISPR to treat the beta thalassemia disease or sickle cell anemia disease. So these two diseases are genetic diseases, and basically there is just a single mutation in those genes that are producing the disease. And then with this approach, we can, or apparently, using this Casgevy drug, we can approach, or we can tackle the problem of these two genetic diseases. So this is a combination of the Vertex Therapeutics from UK and the CRISPR Therapeutics from the States. And this is basically where we are right now regarding the CRISPR technology. So just 10 years after the discovery, we are right now directly trying to treat patients there. So now I will move on to my work related with the CRISPR-Cas system, especially the type 3 CRISPR-Cas systems, and all the work related with this presence of a signaling molecule, which was a ring molecule, and this is what I'm showing in the following slides. And as I have mentioned before, there are two classes of CRISPR-Cas system, and in this case, I'll be focused on this type 3, which belongs to the class 1. And this type 3 is a particular interest because it can cleave DNA, but also RNA. It's the only one that can cleave both DNA and RNA. And if you see here, this is a scheme of the type 3 CRISPR-Cas immunity. This is following basically the first scan I showed you at the beginning, all the 3 steps, the adaptation step, expression and interference. So it's inserting the external nucleic acid into the CRISPR arrays, transcribed, it is processed, and then it's assembled with the effector complex. And then in the interference step, when the second infection arrives, is when it triggers the bacterial immunity. So I've been focused in this part, in the interference step of this immunity in CRISPR-Cas type 3. and so the bacteria can recognize the host RNA and DNA and differentiate it to the external nucleic acid. And then when the external nucleic acid inserted into the cell is when it triggers the bacterial immunity and it degrades the DNA through the effector complexes. But not only that, this is the only system in which it is produced from ATP, this ring molecule called cyclic oligodenylate with different sizes from CA4, CA3, and 6. And these are the responsible of the activation of a secondary bacterial immunity cascade. Basically, most of them are ribonucleases. So to focus on this scheme and link it by the presence of the ring molecule, these are the three interference players in type III CRISPR-Cas system: the effector complex, the one involved in degrading DNA and producing the signaling molecule. Then the ribonucleases, which are the one involved in the degradation of RNA in this case instead of the DNA, and they are activated by this renal molecule. And then lastly, during the last 5 years, it was discovered this family of protein called ribonucleases, which are involved in the degradation of the activator of the ribonucleases and then are switching off the bacterial immunity done by these ribonucleases. So in my case, so these ribonucleases, all of them belongs to the superfamily of CSM6, CSX1, and in my case, I was involved in trying to understand how it was working the CSX1 family. So to do that, we started doing structural studies based on initially in X-ray crystallography. So we tried to get the structure of CSX1 by X-ray crystallography. And well, after a couple of years, it was not easy. We got these crystals that was diffracting decently, it was 2.9. But the problem in this case is that we get stuck at the phase problem. For those that are not crystallographers, the problem we are having in crystallography is that along the X-ray diffraction experiment, we can lose partial information of the structure of the macromolecule we are studying, and we have to recover for different and alternative strategies. And all of the strategies we were following, none of them were working in this case. So we got stuck with this phase problem, and then we realized during trying to analyze in solution how it was this protein, that it was oligomerization, so it was producing an hexamer with this size. And this size is something that we can manage, or is handleable for trying to apply for cryo-EM. So at this point, we thought, okay, let's try to see if we can get at least a low-resolution model from the cryo-EM, and from this low-resolution model, we can combine with the X-ray data, and then we can solve the structure. So that was our idea. And then we combined both data. We started to collect the cryo-EM data in the core facility for integrated microscopy in Copenhagen. And to our surprise, what we obtained, it was this 3.62 Armstrong reconstruction map of the CSX1. And so we were not expecting so high resolution, but taking into account we are immersed in this resolution revolution in cryo-EM, we obtained this high resolution. It was not that high than in the case of X-ray, but it was actually good enough for tracing an initial model and for then combine this data with the X-ray crystallography. And the other information we get from this cryo-EM structure, it was actually the shape of the molecule. So you can see here from this picture that we have an hexamer. It's like a triskelion shape, and it is formed by an hexamer. It's a trimer of dimers, 1 dimer, 2 dimers, and the other 3 dimers. The problem in the cryo-EM data in this case, it was that, as you can see here, this is a local resolution map, and depending on the coloring, is indicating if it is high resolution or low resolution. So this part, which are in red, are from low resolution. So we couldn't trace the full structure of the map of the model using just cryo-EM, and then we had to combine with X-ray data, and then combining with X-ray data, we were able to get finally the full structure of the hexamer, tracing the full model. So this is the homodimer. If we think that this is a trimer of dimer, so this is just one of the homodimer I'm showing here, just to simplify the scheme. So it is composed by a carte domain, HTH domain, and the hep domain. This is the responsible, the hep domain for cleaving the RNA, and this is the one in principle involved the Rossmann fold domain for the recognition of the signaling molecule. So this is the cavity, and this is where we suppose it should be bound, the signaling molecule, and this is the cavity formed in between the two hep domains in the homodimer for recognizing the single-stranded RNA and for cleaving it. You can see here also top view and bottom view of the homodimer. So, okay, we got the structure of the CSX1 in its apple form, and then we were thinking to try to get the structure in complex with the signaling molecule, with the CA4, with the cyclic oligodenylate. And then the problem in this case is that this cyclic oligodenylate is not commercial, so we have to produce it in vivo in bacteria and to purify. So this step was really key for this study. And then if we think about the amount we need usually in crystallography for our structural studies, we have to produce a lot of amount of this for proceeding with our studies. So we got the signaling molecule, and finally we could obtain the complex in between the CSX1 and the signaling molecule. You can see here, this is the 2-card domain, and as we were expecting, the ligand is bound in between in the interface in between the 2-card domain. This is the top view, and you can see the map, the electron density map was good enough to trace perfectly the ligand, the signaling molecule. But not only that, what we observed was that the conformation of the ligand was not unique, so we were obtaining 2 different conformations depending on the incubation, the time of the incubation of the ring molecule with the protein, in a sense that we get this conformation 1 when the incubation was it was low, it was not a longer time, and then with a longer time we got this conformation 2. So these 4 phosphates, which are composed of this ring molecule, 2 out of the 4 phosphates were pointing up in the conformation 1, as you can see here, and then the other 2 ones, it was pointing down in the conformation 2. So it was, we were obtaining finally like 3 different structures. The upper structure, the conformation 1, just when the ligand was sensed into the protein, and then the conformation 2, when the complex was fully activated. So one of the questions we were trying to answer it was, okay, is this conformational chain between the conformation 1 to the conformation 2 transmitting the signal from the card domain to the to the hep domain, so to the catalytic part? And then analyzing this conformational chain, this in yellow is the cyclic molecule, then we observed that all the residues from this card domain to the catalytic side, it was changing, all the residues in this part. So that was meaning that actually there were a transmission of the signal between the card domain to the hep domain. This is what it was happening into the homodimer. But then the other question was to know if, okay, we have an hexamer, which is a trimer of dimer, 1 dimer, 2 dimer, 3 dimer. Is it this transmission of the signal transmitted also to the other 2 homodimers? And then what we did, it was to try to study what is called the hexamerization interface, which is the interface in between the different homodimers. So this is one, dividing this homodimer and the other one here. And what we observed, it was that when the ligand was bound to the card domain, and we tried to analyze all the interaction comparing apoform to the conformation 1 and the conformation 2, all these interactions were changing. And this was meaning that the activation and the conformational chain in the homodimer was also transmitted to the full oligomer, to the hexamer. So this is to illustrate what I was mentioning before. This is the transition between the apo and the conformation 1, and you can see this is the 3 signaling molecule binding site, 1, 2, and 3, and you can see once the ligand is bound to the binding site, then it is trapping the ligand. And also there is a transmission of the signal in each of these homodimer. Actually, you can see that the size of the hole is reducing due to these conformational change. And also, if we see what happened from the conformation 1 to the conformation 2, it was also changing. You can see here how the 2 phosphates that were pointing out were pointing down, and then it was transmitting the signal also to the catalytic part. In this case, the secondary structure, there are not too many changes, but from the amino acid residue point of view, it was huge changes, meaning that it was transmitting the signal to the catalytic part. Something I want to remark here is that the catalytic part, which is here, is all the 3 of them are placed to the same region. So 1, 2, 3, they are pointing to the same region in this case. So most of these CSX1 or CSM6 protein were actually homodimers. So in our case, it was an hexamer, and we didn't understand why it was an hexamer in solution and also behaving like it is. And then the only plausible explanation is to think that probably this protein needs to have an hexamer to be fully active, so it needs to contain a kind of cooperativity, and then we perform some cooperativity assays, and this is the shape of the curve. You can see here that this is basically corresponding with a sequential cooperativity way to function, and also the Hill coefficient was indicating that we need at least 2.5 molecules for the full activation of the complex. And thanks to this information and the different structure we got, we could propose a model for the activation of these RNAs, in which thinking that this is a sequential way to activate. When this first initial ring molecule was bound to one of the homodimer, was transmitting the signal to the other two homodimers. Then the second molecule is bound to the second binding site, and then also transmitting the signal to the other two homodimers. And finally, the third molecule is bound to the third binding site, fully activating the complex. One of the questions that was missing, it was to know if this protein was actually cleaving specifically RNA, because it was in the literature say that these kind of ribonucleases were basically chopping off the RNA, but without any kind of sequence specificity. So in our case, we performed a cleavage specificity assay, and what we found it was that this protein was actually cleaving in between cytidines. And the other remaining question in this case, it was to try to get the complex in between the CSX1 and the RNA. And in this case, we couldn't get the structure either by crystallography or cryo-EM, and we performed docking analysis, and we could propose a model in which this is the hep domain, the homodimer hep domain, the cavity, and then we could propose this model in which you can see how all the different basic residues, arginine and lysine, were pointing to the phosphates of a pentamer of RNA. So, so basically, to conclude this part, it was the first time it was mapped and activated a bacterial immune system, and we found similarities in between this signaling system and the one it is actually in eukaryote, like the RNA's IL system. And we propose also that this protein, this CSX1, could be used as a modular switch for biotechnological application. If we think that this CSX1 is cleaving in between cytidines, so it's specifically cleaving in between cytidines and can be activated by these ring nucleases, this is why we suggest that probably we could use it for this kind of biotechnological application. For example, if we think in the production of nucleic acid detection, like the one it was actually used in during the COVID-19 pandemic. So then we move on to try to study the function of these ribonucleases, how it was degrading this activator of the ribonucleases. And we are starting to study trying to find or to identify those ribonucleases from the same species that CSX1. So the CSX1, we were studying the one from Sulfolobus islandicus, and from the same species we try to identify the ones was working as a ribonucleases in this system, and we identified these two ones, this 0455 and 0811, and we could confirm that it was actually working as a ribonucleases. So this is an experiment in which, when we incubate our CSX1 in complex with the RNA, and we are starting to add different concentration of a ribonucleases, it was actually inhibiting the cleavage of the RNA. So we could confirm that both were working as a ribonucleases. But not only that, when we were checking not just our two ribonucleases, but also this alignment that you can see here, this is the paper, and it was published for the first time, the presence or the discovery of these ribonucleases. What we saw it was that these ribonucleases actually, they appear always by pairs on each organism, like in our case. And that's why we propose that there should be two different subfamilies into the family of the ribonucleases. And according to sequence, we call it then large standalone ribonucleases because it contains a carte domain plus HTH domain, and the other one that were shorter, we claimed that they should be called like small standalone ribonucleases. So according to this, we started to study the structural basis of the degradation of the ring, this cyclic oligodenylate by the large standalone ribonucleases, using cis0811, these ribonucleases, as a representative of this family. And what we did, it was to perform a structural study based on crystallography first. So first we checked the oligomerization state in solution, and we found that it should be a dimer. And this is basically what we obtain into the crystal. So this is the crystal structure of the dimer, and this cavity, it should be the cavity where it's supposed to be bound and cleave the ring molecule. So, and here also you can see from the top view, this is an electrostatic potential map. The blue part is meaning that this is positively charged residues, and this should be the place not just for the cavity but also for the charge that should be bound the cyclic oligodenylate. So then we incubate the cyclic oligodenylate in the presence of our crystals of these ring nucleases, and we could confirm that this ring molecule will actually bound in the interface in between the two carte domain. But to our surprise, what we got, it was not the complex in between the protein and the substrate. What we got, it was the complex in between the protein and the post-catalytic state, because as you can see here in the map, there is no continuous map in these two regions where the arrows are pointing, and this meaning this is meaning that there were a cleavage of this position of the cyclic oligodenylate. And to try to confirm that, we did some mass spectrometry, and as you can see here, this is the control with the CA4 without the presence of the ring nuclease, and then when we incubate with the ring nucleases, we started to obtain this P1 product, which is the 2A2 that correspond to the one we were seeing in the structure. So we also analyzed the structure in its apoform and in complex with the post-catalytic state. And we could see that it was a huge conformational change, not just in the carte domain. We could expect that this is going to happen because at the end it's trying to trap the ligand for degrading the cyclic oligodenylate, but also in the HTH domain, in this bottom part that we are still analyzing what is happening right now. And at the right, what I'm showing here is the active site in the apoform, representing all the different residues that are involved in the binding and also in the cleavage of the ligand. And also here, those residues after the catalysis of the reaction. So you can see here all of them placed in it. So basically, we analyzed how it was working these ring nucleases in the case of the large version of these ring nucleases, and then we proceed trying to study the structural degradation or the structural basis of the degradation of the ring molecule with the other subfamily from the small stand-alone ring nucleases. And we also use the X-ray crystallography for analyzing this, in a sense that what we saw, this is the structure in its apoform and also in complex with the substrate, and this is the domain organization. You can see that here it contains again a carte domain, which is always the responsible of the binding, but it contains a C-terminal insertion instead of the HTH domain, which are pointing in these places, as you can see. And this region is key because it's the way the protein is trapping completely the ligand, and this ligand is completely excluded from the solvent. So there is no contact between the ligand and the solvent, unlike it happens in the other protein where it was completely exposed to the solvent. And analyzing the structure of this complex, what we saw it was that not only we could see, because the resolution was really good, it was 1.5, so we could see all the residues involved in the recognition of the substrate, but also we realized that there were two helices, these two helices that you can see here, that they are fundamental, they are crucial for forming the complex and for triggering the catalysis, not only the binding, but also the catalysis of the degradation of this ligand. Actually, if we remove this interaction, it was completely abolished, the degradation of the catalysis. So we also compare the apoform with the one with the substrate bound form, and again it was a huge conformational chain between the apoform and the one with the ligand bound, and you can see also how it is fully trapped the ligand inside the protein after this movement from this to carte domain. So we described how it was working these two ring nucleases, or two subfamily of ring nucleases, but the question was why it is necessary the presence of two ring nucleases for each organism. And then we started to perform some binding assays and also kinetics assays to try to understand how it was behaving, one and the other. And what we found it was that the small standalone ring nucleases, this 0455, was actually having higher affinity than the other one, but it was degrading in a slower way the substrate. So this means that there is a balance in between the affinity and also the kinetics for what we propose is that probably we need two of them to fine-tune the intensity of the immune response depending on the different infection scenarios. So, and to conclude this part, this is the first time we have shown how it was activated, these ring nucleases, how it was degraded the ring molecule, and also we identify these two subfamilies, the large standalone and the small standalone ring nucleases. And we reveal that this mechanism that is also followed by the phages to escape from bacterial ribonucleases, and this mechanism is it is called anti-CRISPR. And just to finish, I would like to show you that one of the questions that we can think is that if this CRISPR is the only bacterial immunity system in bacteria, and well, traditionally, in the 50s, it was discovered the first bacterial immunity system from this restriction modification system that is has been very useful in molecular biology for their applications. And in the late 80s, it was discovered this CRISPR-Cas system. But what has happened during the last years is that the defense arsenal from bacteria are richer than it was anticipated. And you can see here all the different mechanisms that has been discovered recently. And basically, we can classify all these mechanisms in three. So the strategy follow is when the infection is not that high, so when the phage has just entered into the bacterial cell, the bacteria triggers the degradation of the phage nucleic acid. So, for example, this CRISPR-Cas system and other system that you can see here. But when the infection is larger, the bacteria adopts the abortive infection mechanism, which is inducing cell death. So it's a kind of apoptosis to try to save the rest of the population of the bacteria. And when the infection is extensive, is when the bacteria follow the inhibition of the DNA and RNA synthesis of the phages to avoid the full infection. There are another unknown mechanism, and every month is coming out new different mechanisms, but basically most of them are following these three steps. So why is it important to study these bacterial immunity systems? So, well, there are many reasons, but basically to try to understand how these bacteria defend themselves can be critical for using phages to treat antibiotic-resistant bacterial infection, and also it can be used for manipulate microbiome for industrial application. This is the main reason. And if we think about the huge problem we have right now regarding the antimicrobial resistance, so this is a report from the World Health Organization that was done by the journal The Lancet. About 5 million people every year are dying due to the drug resistance infections. And the prediction is that probably in 2050, around 10 million people will die due to this antimicrobial resistance. If we compare with cancer, this is a lot of people. And so basically, to conclude this growing antimicrobial resistance crisis has revitalized to the research to try to find an alternative to antimicrobial, and the base therapy is one of the most promising one nowadays. And the other reason to try to study this bacterial immunity system is because we cannot forget that this CRISPR-Cas system has been obtained just by basic research, and just by studying this bacterial immunity system, nowadays we can use it as a genome editor. So maybe the discovery of this new immune mechanism can find new tools for manipulating cell and genome. And actually, right now, there are new proteins called prokaryotic argonauts that are starting to be developed for this genome editing application from these studies. So to conclude, this increasing relevance of the bacterial immune system has led to increasing in the discovery and characterization of the phage-resistant mechanisms. And just to conclude, I would like to thank all the people involved in these projects, especially Professor Guillermo Montoya from the University of Copenhagen, and Professor Qinxin Shi from Shindong University in China, and Associate Professor Stefano Stella, who is now in Ensoma Biotechnology as a Vice President, and all the people from the lab who was collaborating with me, Minxia, Ana Luis, Javier, Ricardo, and it was crucial, the role of the people from the proteomic research infrastructure to producing this green molecule for the structural studies, and also it was key, the role of Tillman Pape from the core facility for integrated microscopy to teach me how to handle with sample preparation for cryo-EM, data collection with a small microscope, high-end microscopes, and the data collection and the processing. And I would like to thank also facilities from the different synchrotron Max 4 in Sweden, PSI from Billy and Desi from Hamburg, and the core facility for integrated microscopy in Denmark. Their research support, and just to finish, we are hiring right now. So we have, this is a QR from my group website, and there is currently a PhD offer, so if somebody is interested on that, just contact me. And that's all. Thank you.
So, thank you, Rafa, for this wonderful talk with such amazing structures. So we have time for questions, starting from the maybe younger audience. Any questions, please? One by one. Anybody wants to ask something?
Hola, Rafa. ¿Se oye? Bueno, voy a intentar hacerte la pregunta.
Te digo.
Vamos a ver, me ha parecido súper interesante el sistema, y es un sistema muy interesante para estudiar desde un punto de vista bioquímico. Te voy a hacer unas preguntas sobre la primera enzima, la que
En forma de triskelion,
¿no?
Sí.
Has propuesto un mecanismo en el que la enzima degrada a través del anillo central un RNA, ¿no?
Uh-huh.
Entonces, mi pregunta es acerca de si habéis estudiado la procesividad de la enzima, es decir, si necesita, por ejemplo, enhebrar el RNA o podría actuar de manera, digamos, tocando el RNA de una manera lateral sin tener que enhebrarlo, y si tiene que enhebrar ese RNA, si la procesividad es 5', 3' o 3', 5'. Yo creo que son dos preguntas importantes.
Sí, no lo hemos hecho, pero sí que es cierto que una de las cosas que no entendíamos muy bien era precisamente, claro, esa conclusión de los tres sitios activos respecto a la misma zona tiene que estar implicado con la función. Entonces hemos hecho algún tipo de modelización, pero no llegamos a hacer la parte bioquímica de ver exactamente cómo se producía el complejo, o sea, si realmente necesitaba estar con una determinada estructura, o sea, cuál es el razonamiento de que esté focalizado los tres sitios catalíticos hacia el mismo punto. Evidentemente tiene que ser una cuestión funcional, pero no hemos hecho esa parte de los experimentos, pero vamos, están pendientes.
Y luego otra pregunta un poco de curiosidad. Estos efectores, el C4 poliadenina, ¿hay otros efectores de tipo cíclico de estos?
Sí.
¿Y qué tipo? Es una cuestión de curiosidad.
Bueno, de hecho, esto es un mecanismo que también se sigue en eucariotas. O sea, por ejemplo, el receptor STING, que también está en inmunidad innata en eucariotas, sigue un AMP cíclico.
El AMP cíclico es solo un AMP, ¿no?
Sí, pero hay también variantes en las que existen tricíclicos y existen también tetracíclicos. Lo que pasa es que la mayor parte de los cíclicos sí que es cierto que se han visto en bacteria. Yo he trabajado con el tetracíclico, pero está el tricíclico, que suele actuar sobre una cascada de proteínas que lo que hacen es nicar, no se puede decir, hacer un nicking en el DNA, o sea, cortar una de las dos hebras. Hay otras que lo que hacen es oligomerizar otra serie de proteínas que están involucradas en la replicación. Pero en el caso de eucariotas, lo más clásico es la vía del STING receptor que básicamente lo que hace es una cascada de señalización de la inmunidad adaptativa, y sí que utiliza este tipo de ligandos cíclicos, ya no solamente el AMP cíclico, sino otro tipo de ligandos cíclicos, o incluso el que he mencionado ahí, el RNA System L, también utiliza este tipo de ligandos cíclicos que son de una mayor entidad de unión única adenina, digamos.
Sí, pues sí, a mí sí me... Bueno, ya no, así.
Rafa, me ha gustado mucho la charla. Veo que esto está explotando, todo lo que es todos estos sistemas digamos, de defensa bacteriano, y entonces yo que te quería preguntar un poco por las aplicaciones que esto puede tener en lo que es la terapia génica y demás. Estos nuevos sistemas que se están descubriendo ahora, ¿es que son más específicos? ¿En qué aportarían como herramientas nuevas frente a lo que ya existía o lo que se está utilizando ahora en terapias génicas?
Bueno, en el caso de CRISPR, básicamente he contado un poco en el punto en el que estamos ahora, que es, habitualmente se utilizaban estas Cas9, que es la primera que se descubrió en el laboratorio que utilizaron en el desarrollo en el laboratorio de Jennifer Doudna y Emmanuel Charpentier. Y como he comentado anteriormente, están en estos sistemas argonautas procarióticos también, que lo que hacen es básicamente, o sea, el nexo común entre todas estas herramientas es que reconocen específicamente una región del DNA. Entonces, lo que se busca hoy en día es que ese reconocimiento sea lo más específico posible para intentar evitar que se produzca el off-cleavage, que se llama, que es básicamente tú quieres hacer el target sobre una determinada región, pero que solo afecte a una región del genoma, no que afecte a una región mucho más larga, porque de cara a la aplicación en humanos, obviamente eso es un problema. Entonces, los nuevos sistemas, por ejemplo, los sistemas argonautas procarióticos, que lo que hacen es reconocer aún más específicamente este tipo de regiones, permiten obtener un off-cleavage, digamos, un porcentaje de error muy inferior a los que se conocen anteriormente. Y sin embargo, existen todavía una serie de limitaciones que, por ejemplo, se han podido solventar por el hecho de... Actualmente existen ahora unos sistemas que se denominan CRISPR-Associated Transposon Systems, que estos lo que hacen es asociar la maquinaria del transposón, que es bacteriana, con la maquinaria de CRISPR. Y entonces la particularidad que tiene es que CRISPR actúa en este caso no como una herramienta de tijera molecular, sino simplemente para reconocer específicamente la secuencia, y luego el transposón lo que tiene es la maquinaria para poder realizar el corte, pero no solamente puede realizar el corte, sino que además puede integrar secuencias de DNA de mucha mayor longitud que actualmente se pueden hacer, incluso superiores a una kilobase. Entonces, el problema que hay hoy en día con estas herramientas de edición genética, las que se tienen actualmente, es que se necesita que la maquinaria del hospedador funcione por la propia... por el propio kit que nosotros proporcionamos. Nosotros proporcionamos exclusivamente la tijera y el DNA que se va a reparar, pero la célula es la que tiene que encargarse de hacer esa reparación. Entonces, si nosotros somos capaces de incluir el kit completo en el que tenemos, como en el caso de los CRISPR-Associated Transposon System, la parte de reconocimiento y la parte de corte sin tener que depender de la maquinaria celular, eso es fundamental y eso es lo que se está haciendo actualmente. Utilizar estos sistemas de transposones asociados a CRISPR para mejorar todo este tipo de problemas. Bueno, precisamente el sistema STING del que hablaba anteriormente, o el RNA System, esto viene de bacterias. O sea, lo que nosotros tenemos es una especie de versión evolucionada de todo este tipo de sistemas. De hecho, todos los sistemas estos de señalización en cascada de respuesta bacteriana en eucariotas son exactamente idénticos, en humanos son exactamente no, pero siguen la misma filosofía de una cascada de señalización por un ligando cíclico que permite todo este tipo de degradación de DNA exógeno o de RNA exógeno. O sea que sí, son nuestros ancestros al final y sí existen en eucariotas. Además, una de las cosas interesantes es no solamente estudiar desde el punto de vista de la bacteria, porque claro, estamos fijándonos en cómo funciona la bacteria para defenderse de los fagos y poder intentar utilizar alguna de esas rutas como una futura diana para poder desarrollar un antibiótico, pero tenemos que pensar también en el papel que hacen los fagos o cómo esos fagos, que hoy en día la fagoterapia en general se utiliza como purificando directamente el fago y utilizándolo como un cóctel frente a una infección bacteriana, pero si nosotros entendemos la maquinaria del fago para defenderse de la bacteria, eso también puede ser una estrategia, de tal manera que hacemos un mimicking de esa estrategia para poder atacar a las bacterias que nos interesa eliminar. O sea que es un poco las dos vertientes. Aquí solo he contado la parte de los mecanismos de inmunidad bacteriana, pero la parte del fago es fundamental.
Muchas gracias, Rafa, por la charla, muy interesante. Yo que te quería preguntar sobre esta molécula de señalización cíclica, ¿qué pasa? ¿Que no se puede sintetizar? O sea, porque purificar eso es una pasada de complicado, o sea, de consumo de tiempo. ¿No es sintetizable?
Ahora sí. O sea, ahora hay una empresa alemana que se encarga de hacer todo este tipo de ligandos cíclicos, pero en ese momento nosotros no disponíamos de... Vamos, no existía comercialmente, pero ahora sí. Ahora, de hecho, creo que te producen desde 3 hasta 10, una molécula cíclica de hasta 10 cicloadeninas te las pueden producir. Pero en ese momento no existía y además debe ser muy, muy costoso, porque de hecho el ligando tetracíclico, cuando nosotros lo utilizamos para la segunda parte de lo que os he enseñado, primero nos centramos en las ribonucleases, en la CSX1, esa es la que tuvimos que purificar y producir in vivo, pero luego después, en el caso de las ribonucleasas, nos dimos cuenta de que esta empresa alemana sí que lo estaba purificando, lo compramos y era inmensamente caro, o sea, era muy, muy caro. Entonces sí, ahora actualmente comercialmente se venden, pero debe ser muy complicado la síntesis.
Y no me queda con la copla de la afinidad de esta molécula de señalización, porque, en fin, tú estás expresando esta proteína en bacterias y no iba con ella, con la molécula de señalización, la proteína misma, porque en general puede llegar... Imagino que la afinidad será grande.
Es que nosotros la proteína no la producíamos en el mismo organismo, porque nosotros la producíamos en E. coli, pero lo que realmente sulfolobus islandicus es el que produce el ligando cíclico a través del effector. Entonces tenemos que trabajar, digamos, con dos bacterias, tenemos que trabajar con sulfolobus para la producción del ligando cíclico, pero luego la producción de la proteína en sí la hacíamos en E. coli.
Y oye, otra pregunta, ha salido, creo que lo leí ayer en X o Twitter, que se han descrito haciendo un análisis genómico de no sé cuántos teras de genomas microbianos, 200 sistemas nuevos CRISPR.
Ah, sí, un science paper que ha salido hace poco del MIT, de Feng
Entonces,
Fang,
¿esto ahora qué pasa? Que la comunidad está ahora viendo qué son o sea, porque hay tanta presión en este campo que ahora esto se ha publicado y ahora... ¿qué están haciendo los Stella y compañía, los que están en empresas?
Bueno, claro, el Stella y compañía lo que están haciendo es utilizar las variantes que ellos ya conocen, como por ejemplo, os he contado de Cas9, que es el típico que utilizó Jennifer Doudna y Mario Charpentier y siguen utilizándolo. Él se centra en otro scaffold que se llama Cas12a o CPC1, que tiene una serie de particularidades que le hacen mejor a la hora del corte y más específico en el caso de Cas9. Pero claro, la historia es que están intentando, digamos, por análisis metagenómico, ver cuáles son esas nuevas proteínas que nos pueden aportar algo diferencial a lo que ya se tiene actualmente. Entonces, en el caso del artículo este de Science, lo que están haciendo es ver un poco si existe alguna modificación, incluso ver si son incluso más pequeñas, porque el problema es que luego después esto hay que llevarlo a células. O sea, tenemos que pensar en que toda esta maquinaria tiene que ir insertado en un adenovirus y luego hacer el delivery. Si es muy grande la molécula es un problema. Entonces, está intentando analizar a ver si hay secuencias que son parecidas a estas, mejores en cuanto al rendimiento, pero además encima son más pequeñas y se puede compactar más fácilmente para poder hacer la función de terapia génica. Entonces, eso es un poco la idea del brainstorming de todo.
Muchas gracias, Rafa. La verdad es que es impresionante. Tengo un par de curiosidades de CRISPR como sistema biológico. Si te he entendido bien, la bacteria tiene como una especie de registro de vacunas, ¿no? Tiene incorporados...
De previas infecciones.
Eso es.
O sea, el CRISPR-Array es simplemente trocitos. Digamos que tiene un sistema que lo que hace es trocear el ácido nucleico que le invade y lo guarda en una especie de recordatorio que se llama CRISPR-Array. Entonces, él ya lo tiene ahí y lo guarda para cuando existe una segunda infección, dice, ah, bueno, este ya sé quién es.
Ahí voy. Entonces, si guarda un pequeño fragmentito...
Sí.
El genoma viral es, bueno, varias kilobases, por ejemplo. Entonces, ¿por qué ese trozo?
Vale, porque lo identifica, o sea, existe como una especie de marcador que es capaz de identificarlo la propia bacteria y sabe cuál es el trocito exactamente que tiene que cortar. Eso es básicamente el trabajo que hizo Francis Mojica con todo este tipo de bacterias. Entonces, esos clústeres repetidos que se insertan, la bacteria es capaz de reconocer dónde está una señal que se llama PAM, y esa es la señal que le sirve a la bacteria para decir, bueno, este es el trocito que tengo que cortar y meter dentro del CRISPR-Array.
Y dentro del genoma viral, ¿dónde está ese trozo? O sea, ¿pertenece a una zona codificante?
No.
¿Una proteína? ¿Una zona de regulador? Yo no
Suelen
sé,
ser
o
zonas
sea, ¿hay algo sistemático en eso?
Suelen ser zonas reguladoras, suelen ser zonas de
Reguladores.
DNA basura, digamos.
Ajá.
No era del todo funcional.
Ya. Oh, qué interesante. Y hay fagos de RNA. ¿Qué pasa con ellos? ¿También actúa CRISPR contra ese RNA?
Actúan los sistemas procarióticos de argonautas, estos que he contado así un poco de soslayo, que es lo que están utilizando hoy en día como nueva herramienta de edición genómica.
Ya. ¿Y qué diferencia, cómo distingue un DNA propio de otro no propio, la bacteria?
Bueno, en el caso de los sistemas tipo 3 es por hibridación, porque precisamente cuando genera el CRISPR, cuando se transcribe...
Quiero decir, al principio de todo, imagínate que no ha sufrido una infección previa de un fago nuevo. Ese DNA que tiene que haga ver a la bacteria, coño, esto es de un virus.
Realmente lo que dispara la señal de la respuesta inmunitaria es cuando llega a la interferencia y se da cuenta si es suyo o no es suyo. Antes solamente lo que hace es acumularlo, no lo sabe.
Bueno, gracias. Muchas gracias.
Bueno, no hay ninguna pregunta más. Yo creo que ya podemos agradecer a Rafa por esta fantástica charla. Gracias.
Gracias. El paso de la entrega de la taza. Sí, tengo 3 versiones, yo creo, porque llegué, me dieron la del